Heptahelical receptors, ligand recognition, and conformational dynamics in biology.
Thank you. I'd first like to thank the organizers for inviting me to this meeting and for inviting me to give a talk. Before I start the scientific portion, I wanted to make a brief comment about my period in Gobin's lab. I love timelines. And last night I was thinking about how to present this, and after hearing the talks yesterday, I decided to make a timeline that represents roughly the period of time of Gobin's career when he first started accepting people into his lab in Vancouver in '54, extending to the last new person to come in probably 2007. This is my period here, July 1st, 85 to August 1st of 90. And the list of names on this slide is actually the list of people who overlapped with me at MIT in the lab during that period of 5 years. They're not really in any particular order. They're roughly in chronological order as I could remember them, and I had to go back and fill in a couple of people that I forgot. I think this is a complete list. The people here with stars, Dan, Sadhu, Haibao, Roland, and Bob, are the people who I wrote papers with as co-authors during this period of time. And I just want to thank Gobin for accepting me into his lab, for introducing me to this amazing group of scientists during a very intense period of my scientific training. Roland is my best friend, and you don't need a best friend too often. And in this environment, it tends to bring people together. I also wanted to just point out that in my particular case, my path to scientific research was a little bit different than many others on this list, since I came to Gobin first by coming across the Charles River from Mass General Hospital. I was a resident in medicine doing my clinical training, and I intended to do clinical fellowship training at Mass General. But instead of working in a lab during my clinical training, I decided to take the unusual step of breaking away from that system, which I considered to be suboptimal, at least for me. And I came to Gobin and I asked him if he would accept me into the lab. I had had some training in biochemistry at the University of Chicago with Paul Sigler, Tom Kaiser, and Bob Heinrichsen. So he did accept me. The Mass General folks said, well, if you really want to, you can go to MIT. We'll save us a place for you. And in fact, they're still saving the place. John Potts, who was my chairman, expects me back anytime to continue my clinical training, which I never went back to. So thank you, Gobin. Now, with respect to the scientific portion of the talk, I'd like to continue roughly on the same vein that Wayne Hubbell was talking about, which is technology development. Although the kind of technology that we're developing is related to our goal of now studying the dynamics of GPCRs in the membrane environment in a single molecule regime. In order to do that, we have to develop technology that is mainly designed to provide labeled material at high fidelity so that we can either reconstitute it or study it in its native membranes. And as opposed to the strategy that Wayne was discussing, this sort of site-directed mutagenic method, which has been useful up to this point, we wanted to develop a new method that I'll tell you about for labeling receptors. Now, why are receptors important? This is a complicated slide, and I invite you to take a look at this article by Overington in Nature Reviews Drug Discovery, 2006. It's a fantastic summary. But we hear a lot about the issue of GPCRs and their relevance to pharmaceuticals. And people say, well, 50% of drugs, 40% of drugs, and so forth. But the real number is here in the middle. This is really fascinating. For all approved drugs, there are only 266 human targets. Many drugs target pathogens, for example. Many drugs target non-human targets. But of the human targets, there are only 266. And of those, a remarkable 27% of that number, 266, actually target GPCRs that are in the family 1 or rhodopsin family of receptors. This is far outweighing the next category, the 13% here, which are all channel targets. Now, if you believe Sir James Black, basically, who contends that if you want to discover a new drug, you should start with an existing drug, then there's a lot of work to be done here in GPCRs. I'll just also mention in passing that of the 726 drug GPCRs that are encoded in the human genome, probably about 100 now are still orphans. Many of those are CNS expressed. So there are still a lot of additional space for drug discovery. Now, why is this problem difficult? The problem is difficult for a variety of reasons, some of which I mentioned here. First of all, GPCRs have no modular structure. You have to study the whole thing. You can't express a portion of it and get it to function in any meaningful way. Secondly, although detergents are very useful to reconstitute, membranes are really required to understand the full function. That's a technical problem. Thirdly, there are no orthologs in lower organisms of GPCRs, and therefore these GPCRs can't be expressed readily, say, in E. coli for overexpression. They must be expressed in eukaryotic cells. Also, GPCRs as receptors exist in low abundance in nature. Finally, I'll just make one point, considering the historical context here. The formalisms that the pharmacologists used to study these receptors really were developed 50 years ago, and almost exactly 50 years ago. If you want to pinpoint the time, you would look at a paper by Robert L. Stevenson, 1956. And in that paper, he built on earlier work. The term receptors had been used for decades, but Stevenson pointed out two terms that we now use in continuing in this field. One is affinity, and one is efficacy. He also pointed out this term stimulus. He defines stimulus to be the event that happens when a drug binds to a receptor, but before the output can be measured. Stimulus. And that's really what we're studying. That's what Wayne was talking about, and that's what we're interested in. That was described in 1956 by Stevenson, and we're still trying to figure out really how it works. Now, we've developed a number of technologies. One is computational methods. This is really possible by the fact that we now have reasonable crystal structures, and we now have reasonable computing technology with the proper force fields and so forth to do all-atom simulations. And we've spent a lot of work doing all-atom simulations of GPCRs in the last few years, mainly led by Thomas Huber on that recently. Just to give you a little follow-up on what Wayne said about the dynamics. This is a simulation, an all-atom simulation, where I'm just showing you a few of the phospholipids and a few of the waters and ions. This is the beta-factual engineered T4 lysozyme on the cytoplasmic surface. But this is basically how the receptor is moving. This at the time was a 180 nanosecond simulation, which was one of the longest ever done at that time. It's now been far outstripped by the David Shaw group. But one thing I want to point out is that this structure is moving, and in fact the carazolol, which is an inverse agonist here in the ligand binding pocket, also moves substantially during this simulation. Interesting. Now, we've done also, in collaboration with Merrick and Rennigan, coarse grain simulations of rhodopsin in a bilayer. This is basically how rhodopsin is moving around if you look at from the side view and the top view. And you can do a formalism where you create these coarse grain models and you put, say, 16 of them in a membrane grid, then you start the simulation and see what happens. And this is very instructive because if you just run the simulation, you see that over time, these receptors in the bilayer tend to oligomerize. The role of dimerization and oligomerization in these receptors is not fully understood, but we think that oligomerization probably happens due to the physical interaction between the protein and the bilayer. And we're still working on this question. Now, the second technology that we developed, and that we're developing, I alluded to earlier, this is a method to generally put a label, say a fluorescent label, at any point in the GPCR. We don't want to use the cysteine chemistry, the cysteine mutagenesis method, because it's cumbersome if you want to study receptors which aren't as well understood as rhodopsin. For example, in the case of rhodopsin, you have to remove a lot of cysteines before you put back the cysteines that you're interested in. Here, about 6 years ago, we started trying to develop an amber codon suppression method to do unnatural amino acid mutagenesis in GPCRs. As I told you before, they can't be expressed in E. coli. We were aware of the work of Peter Schultz and admired that greatly. But Peter Schultz's methodologies are primarily engineered to work in E. coli. There was some indication that maybe this general method of amber codon suppression might be adaptable to even mammalian systems, and we were anxious to move forward with that. In this method, we were trying to introduce amino acids that had keto-containing groups, either acetyl phi or benzoyl phi. Later, we also tried to introduce azidyl phi into rhodopsin, expressed in the membrane cells. But this is a non-trivial problem, and we're using amber codon suppression, which I will assume you know about, and this particular paper was actually referred to yesterday by somebody. Now, our problem, we ran into problems very early on with this. And our first problem was getting the correct engineered tRNA. We were pretty sure that we knew how to engineer the tRNA. What we didn't know was how to express it. So when we ran into problems, we first went upstairs to Marka Konarska's lab, who used to be a postdoc with Phil Sharp, and we got her to help us to actually measure the expressed tRNA levels that we had in our cells. When we figured out that our tRNA expression was poor, we turned to Tom Rajnandar for help. And Shishin Yi in our lab and Thomas Huber visited Caroline and Tom at MIT, and I went up there a few times. And we finally figured out a way, with Tom's help, to get high-level expression of our engineered tRNA in HEK cells. And we monitored this process, this painstaking process, by using a luciferase reporter assay that Tom developed. And I won't go into detail, but we could actually optimize the expression of the engineered tRNA, the suppressor. Secondly, we needed to engineer an amino-acyl tRNA synthetase that would recognize the unnatural amino acid of interest and be orthogonal to the tRNA. This was easier because there was a crystal structure available by the time that we actually started carrying this out. And we went through a number of iterations and came to these two mutants of the tRNA, this one for acetyl and this one for benzoyl phi. But we had to optimize for orthogonality. And again, we used, in collaboration with Tom, the luciferase reporter assay. And I won't go into detail. But we could show that we had an orthogonal pair. We were encouraged enough to move forward with trying to actually get this to work with GPCRs. And the GPCR that we're interested in is a chemokine receptor called CCR5. CCR5 is here. It's a rhodopsin class GPCR. If you put it side by side with rhodopsin, most people wouldn't be able to tell the difference. In this experiment, we express the CCR5 in HEK cells, and we put an epitope tag at the C terminus. This epitope tag is the 1D4 monoclonal antibody epitope. It's at the C terminus of the protein. Here's the wild type expression. This is a Western blot where we're detecting with 1D4. We now make a mutant at phenylalanine 2,6-C. We introduce amber codon. That's at this position. If we co-express the mutant and the tRNA, we see nothing here, as we did when we introduced the amber codon. That's because we get termination at the amber codon. It doesn't read through to the antibody epitope anymore. Now, if we co-express three elements, the mutant gene, the tRNA, and the synthetase, we still see nothing on the western. It's only when we add the unnatural amino acid to the medium, it gets taken up by the cells, that we see this nice band that corresponds to full-length CCR5. That's telling us that we're getting read-through of the full-length product. And we presume at this point that acetylphenylalanine is at position 260. This is a very cool result. But we can take it further because we can assay in the same cells for activity. Here we use a calcium flux assay. Here's wild type. Here's condition 2, 3, 4. With condition 5, where we're expressing the three elements and adding the unnatural amino acid, we get 32% of the wild type level of calcium flux. This is phenomenal. This is an amazing result for this type of an experiment. Now, we're trying to, this is a mutagenesis experiment, but we're trying to get a functional protein. Because what we want to do is put a probe at an informative site that's near some site that we're probing, but doesn't interfere with the function. And in fact, we can take, doing these dose-response curves with calcium flux, we can do a classic dose-response curve with RANTES, which is the agonist ligand, and show that this mutant, phenylalanine 260 to acetylphenylalanine, is wild type in every respect. But now we have a keto group on the outside of the cell. Cells are supposed to be essentially ketone negative. I won't show you these fluorescent traces. Now we've gone on to make orthogonal pairs for acetyl, benzoyl, and azido feed. These are all for chemical labeling purposes, but benzoyl and azido can also be used as cross-linkers. These are photoactivatable cross-linking reagents. And the cross-linking advantage here is that you know at the beginning of the experiment where the cross-linker is on essentially your bait protein. So you do the cross-linking. You don't have to spend time searching for the location of the cross-link. You just determine that the cross-link happened or not. And you can scan through regions of your proteins of interest. Interestingly, we also had an interest in the azido as an infrared probe. Just one slide talking about that. The reason this is cool is that azido is an environmentally sensitive probe based on starter fat. But in an infrared spectrum, here's all the action happening in the protein. We've worked a long time with Fritz Siebert in Freiburg on different spectroscopy of mutant rhodopsins. And we've assigned a number of difference bands where we do light versus dark, IR. This is a different spectrum. Here's the protein. The azido is way out here. This is the azido absorption. There's nothing essentially in this region that's related to the protein. So we can put these azido groups at different locations on the receptor and interrogate as a function of state, off or on. And here's an example of azido at one position, 102. There's no difference in the two states. Whereas at, say, 250 here, this is the site that Wayne was talking about earlier, this cytoplasmic loop region, or 227, we see differences in the environment of the azido. It's a little bit difficult to exactly interpret what the change in environment is with azido, but it's a proof of concept experiment, and we're very excited about this. And by the way, this is a direct proof that we actually have the unnatural amino acid in the protein. Now, of course, we'd like to do chemistry on these, and this is what we're doing now. We've just finished this work. We've done the hydrazone ligations with the keto-containing groups, and starting with Bertozzi and the click chemistry here. And we can get fluorescent labels now generally in GPCRs, and in CCR5 in particular. Now, Wayne talked about his genesis strategy. We're doing the same thing using our unnatural amino acid method on CCR5 and CXCR4. It's close relative. And we're targeting now the cytoplasmic surface, These are sites where we've introduced amber codon. The way we do these experiments generally is to make a mutant to phenylalanine and a mutant to the amber codon. This is cool because once you have the amber codon mutant, you can introduce, in principle, any unnatural amino acid at that position for which you have the orthogonal pair. So far we have actually 4 because we've also done methoxy. So we could put methoxy, azidyl benzoyl, or acetyl feed at any position, any unique position here. So that's one unique label where we can recover essentially quantitatively. Now I told you earlier that we'd like to do single molecule studies. How are we going to accomplish that? Eventually we think that this technology will work in live cells. The labeling chemistry is a little bit tricky, but we're trying to get the conditions so that we can label the surfaces of cells and then measure in real time single molecule events. But until then, we worked out this interesting compromise. We have a turf single molecule setup that we built in the lab, and we developed this little microfluidic chamber. In this chamber, we have a cover slip here that we drill holes in and put tubes here, and we have a glass slide at the bottom that's sealed. If you look now at this surface blown up, we can put on this surface a collection of molecules that we absorb here that we actually build within this surface. So we flow one at a time, the biotinylated DSA, streptavidin, the biotinylated antibody to orient the membrane, and then the phospholipids in the presence of expressed chemokine receptor, CCR5, that has the epitope here. So what we end up with is a tethered bilayer that has receptors oriented in that bilayer. There's no detergent, it's a real bilayer. We've shown that and we spent a lot of time with that. It's amazing that this structure self-assembles in this chamber, but it does. Now this experiment, and this is my last slide, describes our first single molecule experiment that we carried out recently. In this experiment, we use the apparatus I just showed you. And here, in this experiment, we have the recombinant CCR5 in the tethered oriented bilayer on the chip that's within the turf range so that we can see a fluorescence event that's happening on the bilayer. In this case, as a proof of concept, we're adding a labeled ligand, MIP1 alpha, that's fluorescently labeled. Here the receptor's not labeled, the ligand is labeled. And we hope to measure single molecule binding events. This tethered bilayer is extremely stable, so we can flow the agonist ligand into the chamber, wash it out, flow it in, wash it out, and so forth. And in this experiment, we're doing that with different concentrations. 0, then 1 picomolar, 0, 10, 0, 100, wash, 1,000 picomolar of the agonist ligand that's labeled. And so what you'll see here is we're flowing now. You see a little puff of fluorescence, and then there's a washout, and then you'll gradually see more and more as we get above. That's 100 picomolar. And now at 1,000 picomolar, each of these dots are individual binding events which then decay over time. So it's relatively trivial to measure off rates. You can derive off rates. You can derive other kinetic constants from this experiment. But for us, it's a very exciting result. And we also now can put our fluorescently labeled receptors into the bilayer. And in the same type of an experiment, do the equivalent of single molecule FRET in the system. So we hope to push this to the limit in future work. This is my lab at Rockefeller. Especially I'd like to acknowledge Thomas Huber right here, who did the computational work and some of the single molecule work. Thank you.